Well, hello! So we've taken a look at the path of blood flowing through the mammalian or bird heart. We've taken a look at the heart cycle. So we've tracked the process of different pressure pulses contributing to squeezing blood through the heart. So now we're ready to talk about how contractions in the heart are coordinated. And to appreciate this, let's just recognize that the heart is an electrical organ, right? So and here with a heart is George the cat, by the way. Hopefully he won't interfere too much. Anyhow, the heart is an electric organ, right? So we can actually detect the electrical signals produced by the heart by attaching EKG or ECG leads at different places across the body surface, right? And the electric signal that the heart produces is strong enough that we can pick that up and then measure it and use that information for diagnostic purposes. So what this means on a cellular level is that there are small local changes in membrane potential that can cause voltage-gated ion channels to open suddenly and lead to rapid depolarization. So this should remind you of what you've learned in the past about how nerve signals are transmitted. The process is pretty similar across heart muscle cells, although there are some distinctions. Right, so the depolarization across the heart muscle cell surface stimulates the contraction. And we'll start investigating this more closely when we talk about the control of muscle contraction, skeletal muscle contraction, towards the end of the semester. So this is kind of a preview of that topic, and we're not going to cover similarities or differences in cardiac depolarization compared to skeletal muscle, but that will give you a good framework for understanding cardiac muscle function. So the origin point for depolarization leading to heart muscle contraction is a cluster of cells that are known as pacemaker cells that are found in a location known as the sinoatrial node. In mammals, these cells are myogenic cells, so they have their developmental origin as muscle cells found in this location. And they have a unique property in that they can spontaneously depolarize, right? So if you think about a neuron's action potentials generally don't just depolarize and start firing on their own, right? They're dependent on inputs that summate that then lead to production of the action potential. So in contrast, pacemaker cells, they have the transport proteins, right, to establish an electrochemical gradient, right, for the purpose of depolarization, but they also have a set of leaky ion channels that will lead to a spontaneous depolarization. And this image here comes from our textbook. This is an alternate view of the heart, just highlighting the structure of some conductive fibers that are found across the surface of the heart. So the signal originates at the sinoatrial node. It then propagates across the atria, but it also spreads along these conductive fibers down to the ventricle, down to the tips of the ventricle. And these conductive fibers then also facilitate spread of the depolarization signal upwards from the ventricle. The other thing that I want to highlight that our book doesn't do a very good job of highlighting is the point that there's actually a layer of fibrous connective tissue between the atria and the ventricles that is not conductive. Right, so this layer of fibrous connective tissue insulates the signal in the atria from the ventricles. So that controls the nature of how that depolarization signal gets conducted. So here's another cartoon just to illustrate how depolarization starts and then spreads over the course of a heartbeat. And here we're actually starting at stage 5 of how I outlined the heart cycle, atrial systole. Right, so the signal will originate at the sinoatrial node. It will then be conducted across the heart cells, so that will lead to contraction of the atria, right, both of the atria together. But then it's blocked, right, at the fibrous tissue layer between the atria and ventricles. It will reach the atrioventricular node, right, so this is part of those conductive cells, and from the atrioventricular node, the signal will then be propagated down to the tips of the ventricle first. So the atria squeeze from the top down, right? That's helpful for moving blood from the atria down into the ventricles. The ventricles then, thanks to these conductive fibers, will start to squeeze from the bottom up, right? And that's helpful for the purpose of emptying out the ventricles, right, into the aortas, right, producing force in that opposite direction. And it's possible, again, to measure this uneven charge distribution in other body tissues. So you don't have to actually go in and measure the heart directly. And I think we're generally pretty familiar with the readout of this depolarization, repolarization signaling process, right? That is an EKG or ECG, electrocardiogram. And we could spend an entire course talking about the structure of electrocardiograms and how to interpret different things that can go on depending on what's happening with conduction across the heart. But we're comparative animal phys, so we don't really have time to do that. So we're just going to look at a classic example of an ECG just to appreciate kind of how this uneven charge distribution shows up on the ECG. So we've got time on our x-axis, we've got the electrical potential difference on our y-axis, right? Initially we'll see atrial depolarization, so that comes across as our P wave here. Then we'll have repolarization, and you'll notice ventricular depolarization produces a much stronger, faster signal as our R wave, and then things conclude with ventricular repolarization towards the end. One thing we saw in lab is that the action of the heart can be modulated by other factors. So for example, there can be global signaling factors or hormonal signaling factors, like if you see a bear and that triggers a release of epinephrine or adrenaline, right, that's going to circulate in your bloodstream, and when that reaches the heart, that can lead to an increase in heart rate and heart stroke force. There are also direct nervous system inputs to the heart, right? So you can just think, and if that changes inputs, sympathetic and parasympathetic inputs to your heart, that can change heart function as well. So for example, the sympathetic branch of the nervous system input can lead to an increase in heart contractile speed and heart contractile force, right? That's illustrated here at these sympathetic cardiac nerves. And then in contrast, parasympathetic inputs, primarily through a cranial nerve known as the vagus nerve, right, can lead to a decrease in heart rate, decrease in heart stroke force, right, so that energy can then be used for other functions instead. One other source of control that we have not talked about yet is this third form of intrinsic control, right? So this is not due to inputs from other sources, not due to the activation of pacemaker cells, but just the process of stretch of the heart muscle can lead to an increase in contractile force. So this process of stretch increasing contractile force is referred to as the Frank-Starling mechanism, after the people that originally described it. And this property of stretch leading to an increased contractile force is actually also true for skeletal muscle. And so again, we'll look at that in greater detail when we talk about skeletal muscle function. But essentially, the thing to realize is if a larger volume of blood returns to the heart, right? So that's what can happen, when you start to exercise, right? Your skeletal muscles are squeezing your veins, right? That is pushing additional blood faster back to the heart. So for each heart contraction, there can be a larger venous return of blood to the heart, right? So if there's a larger volume of blood in the ventricle, that's going to stretch the ventricle further, and that's going to directly lead to an increased contractile force to squeeze all of that blood back out of the ventricle. This can be a tricky concept to really grasp, so I think it's helpful to see a couple of different models of circulatory function to help us kind of appreciate and understand how this works in general. So in the center is probably what you might initially think about when you think about a closed circulatory system, right? You've got a pump, it's pumping blood through microcapillary beds or vascular beds here, then that blood returns to the veins, and then it returns to the heart. Okay, but in contrast, we know that veins are distensible, right? So it's possible for more or less of the total blood volume in a person to be found in the venous portion of the circulatory system. So a contrasting depiction is on the left here, this open model of circulation, right? So here we've got a pump pumping through the vascular bed, then we've got venous return, and this is just illustrating kind of infinite capacity in large veins, and that return of blood to the heart, right, is really controlled by the extent of venous resistance, right? So when at rest, resistance is going to be higher. That's going to slow the return of blood. When exercising, resistance will drop, and that will lead to a faster return of blood to the heart. The reality is more likely somewhere in between the open and closed models of circulation. So if we just think about veins being distensible, venous resistance will contribute, but this is a closed system, so there isn't an infinite capacity found in the veins. But hopefully this helps you better appreciate and understand how you could have an increased venous return of blood to the heart, particularly during exercise. That will lead to stretch, and then that will lead to increased contractile force to deliver that blood back out into the circulation. The last thing with this topic is you do need to be aware that not all animal heartbeats start from a muscle cell. So as a contrasting example, here is the heart structure of a lobster, and lobster hearts are under neurogenic control, right? So that contrasts with the myogenic control we see in the mammalian heart. So this is a cluster of neurons that will fire, right? And that will just lead to a contraction. Lobster hearts do have similar sympathetic and parasympathetic modulation of heart function, right? So they could have an increase or decrease contractile force and frequency. But the stretch mechanism for a lobster heart is a little different from the Frank Starling mechanism I just showed you. Specifically, if the lobster heart tissue is stretched, this cardiac ganglion, so this cluster of neurons, will then start to fire more frequently and more intensely. And that will lead to an increased contractile force and increased contractile frequency for this heart tissue. So this is distinct from what we see in mammals. It's actually a little closer to what we'll see when we look at skeletal muscle function. But again, for our purposes, the main thing to just be aware of is that the control mechanism can depend on the specific animal that you're talking about. And there you have it, heart control.